I'm David Pellman. I want to thank Adrian and the other organizers for the kind invitation to this wonderful meeting. It's a real honor, and I'm pleased to be able to present the work from my group. I'll share my screen. Okay, these are obligatory disclosures, none of which impact my talk today. So the general question that I want to address today is how during tumorigenesis, cancer cells go from a normal genome with 46 chromosomes to a highly complex cancer genome. Cancer genomes can have hundreds, thousands, or even tens of thousands of genomic rearrangements. In some senses, we used to think that we understood how this worked, which was meaning that it would go by classic Darwinian gradual evolution, with small-scale genetic changes occurring one step at a time, one generation following the next, eventually piling up and giving you the final genome complexity. However, you could wonder how long gradual evolution would really take to get the typical complexity seen in many epithelial tumors. And a new idea has emerged in the field that my group's been very involved in, which is that in addition to gradual evolution, genome evolution can proceed in episodic bursts, where a small number of catastrophic events can rapidly generate this kind of complexity. One mechanistic theme that I'm going to emphasize in this talk is that these catastrophic bursts can oftentimes originate from defects in the architecture and the integrity of the nucleus. This is a DNA stain of sort of extreme example cancer cell line, and what you can see is highly abnormal shapes of the nucleus. That's shown here as abnormal lobes, little nuclear blebs, structures that we're going to talk a lot about called micronuclei, and other structures that we'll also discuss in great depth called chromosome bridges. Nuclear atypia in cancer has been noticed ever since the late 1800s, and in the late 1920s, pathologists appreciated that the degree of nuclear atypia could predict patient prognosis, and to this day, it's still an important component of assigning tumor grades. So the biological significance of cancer nuclear atypia has been known for a long time, but what has been much less clear are what are the actual consequences for nuclear functions for defects in the architecture of the nucleus, and that's something that I'm going to show you some of our data on. So there are at least three major processes that are known to drive rapid genome evolution. The first is the duplication of the entire genome, which can occur from cytokinesis failure, doubling chromosome number, doubling centrosome number, generating a lot of genome instability, and that's something that in 2005 we showed could generate tumorigenesis in a mouse model. And we've studied this for a long time. In this talk today, I'm not going to discuss whole genome duplication further, but I will emphasize that it's now known from cancer genome sequencing that whole genome duplication occurs in something like 40% of all human cancers. The topic for today is going to be first a review of our work on a newly discovered catastrophic mutational process called Chromothripsis, short for, or a Greek neologism for chromosome shredding. And in the second half of the talk, I'm going to talk about another genome instability phenomenon called chromosome breakage-fusion-bridge cycles, and I'm going to specifically address the relationship between these two processes in my talk today. And the work on BFB cycles was recently published in this paper from Neil Umbre from my lab. Okay, so Chromothripsis was first discovered by Peter Campbell and then rapidly identified by many other groups And what was unusual about it is massive rearrangement that was curiously restricted to only one or a few chromosomes. We normally think of mutagenesis as impacting the genome homogeneously, and how you could get massive rearrangement on only a few chromosomes was unclear and interesting. We now know that, like whole genome duplication, Chromothripsis is very common in cancer, with frequencies that range between 20 and 65% in the common epithelial tumors where there's a lot of genome instability. And for example, in pancreatic cancer, it was proposed to account for up to 50% of all the chromosome rearrangements in these tumors. Although first described in cancer, Chromothripsis is not specific to cancer or to humans. It's a general mutational process. It can cause rare human congenital disease. It can be passed through the germline, and in principle could therefore contribute to organismal evolution, a point that's underscored by the fact that it's now been described in a diverse organisms, including plants. So a number of years ago, based on our finding that chromosome segregation errors that generate these abnormal nuclear structures called micronuclei could lead to extensive DNA damage, specifically of the chromosome in the micronucleus, we made the proposal in 2012 that micronuclei could be a source of Chromothripsis. And to address, of course, this audience, more than many others, will fully appreciate that Chromothripsis is a genome phenomenon. You can't assay it without doing genome sequencing. So in the 2012 paper, the hypothesis really came from classic cell biology techniques, but we realized that we needed to do better, to be more direct. And what we then did was to develop a procedure that we playfully call look-seek, where we could create micronuclei de novo with no pre-existing DNA damage. And work from others has shown that the micronuclei have fragile nuclear envelopes that break and then develop DNA damage. So we could make the micronuclei, watch the envelope break, see surrogate markers of DNA damage, let the cells divide and see the micronuclear chromosome then be reincorporated into daughter cells. And the way we do this experiment now is to then come in with a microcapillary hooked up to a robotic arm so that we can come into the culture, isolate these single cells, and subject them to single cell whole genome sequencing. There's a lot of interesting bioinformatics and methodology involved, but I do want to highlight that we have haplotype resolved DNA copy number measurements for these experiments that can unambiguously identify the chromosome that was in the micronucleus. And furthermore, I want to emphasize that by comparing the two sister cells, we get important validating information that is the essential equivalent of a biological replicate. Okay, this work is mainly published. I'm not going to go through the primary data, but I'm going to show you a short animation that summarizes this work. So micronuclei can originate from mitotic errors, which in some cases can originate from too many centrosomes in cancer cells. This creates abnormal chromosome attachments that cause chromosomes to lag in anaphase and then be mispartitioned. When the nuclear envelope reassembles an interphase, you can get a micronucleus. The very same thing can happen if you have a single unrepaired chromosome break that gives you an acentric chromosome fragment that goes into a micronucleus. We and others showed that these micronuclei undergo very poor DNA replication. The envelopes are fragile and prone to spontaneous disruption. Somehow, the exposure of the chromosome to the cytoplasm leads to DNA damage and chromosome fragmentation. And if the fragments are randomly distributed to the two daughter cells, and we come in and sequence them, essentially what we can see is that this experiment recapitulates all the known sequence characteristics of Chromothripsis in cancer genomes. So what you're looking at here is the haplotype copy number of the affected chromosome from one telomere to the next, and you can see that fragments of that chromosome, which is under-replicated, are either present or absent, or present or absent, giving you the characteristic so-called oscillating DNA copy number pattern of Chromothripsis. The fragments are then randomly joined together, and most compellingly, if you look carefully and compare the two sister cells, you can see that if a fragment is retained in one daughter, it's missing in the other, and vice versa. And this mirror image copy number pattern really gave us great confidence that we were looking at true rearrangements and alterations to the genome. Okay, so now for a mechanism for Chromothripsis, we can start to simplify. And we can say that one first step would be to form a micronucleus. The next step, which has a lot of interesting biology attached to it, is that the nuclear envelope on these structures is fragile and ruptures. I won't have time to discuss it in detail, but we've recently elucidated at least the beginnings of the mechanism to explain this nuclear envelope fragility. But the key point for this talk is that during mitosis, the lagging chromosomes undergo defective nuclear envelope assembly and lack nuclear pore complexes, leading to defective nuclear import and a lot of downstream consequences like poor DNA replication. Even at the outset, because of this poor replication, I'm going to show you evidence that there's a starting low level of Chromothripsis, and then the envelope breaks, giving you more DNA damage, which gives you more Chromothripsis. And then I'm going to show you more evidence that these under-replicated chromosomes, upon entry into mitosis, undergo a burst of abnormal DNA replication in the middle of mitosis, which then further adds to DNA damage, chromosome fragmentation, and accentuates Chromothripsis. So the formation of a micronucleus leads to a whole downstream cascade that promotes Chromothripsis. Now, in the next part of my talk, I want to address a general question of whether different abnormalities in the architecture of the nuclear envelope lead to different mechanisms for Chromothripsis, or is there a general common mechanism? And we're going to start, we're going to address what happens in chromosome bridges, which look very different from micronuclei. But although there are some differences, I'm going to propose to you that essentially a very similar cascade occurs in chromosome bridges that will lead to this kind of mounting accumulation of Chromothripsis and ongoing genome evolution. Okay. Chromothripsis, chromosome breakage-fusion-bridge cycles have long been considered major genome instability phenomenon in cancer, and have long been considered a major origin of oncogene amplification. The concept behind this was proposed in the late 1930s and in this paper in 1941 by the famous Barbara McClintock. And McClintock appreciated the following things could happen if you have a broken chromosome N. So if you start, this is showing a chromosome in interface with a centromere and a chromosome arm here, and we're going to imagine that the end has been broken, which could happen in cancer if you have critical shortening of the telomere, or it could happen by a variety of mechanisms that would lead to spontaneous DNA breakage. And what McClintock appreciated is that if this broken chromosome is replicated, you'll have two break ends then next to each other, which can then be ligated together. And what this will cause will be the formation of a so-called dicentric chromosome, and the two centromeres will be pulled between the two daughter cells. So the bridge will form in mitosis and persist into the next interphase. How these bridges are then actually broken has been a major topic in the field that's been surprisingly poorly understood and much debated. But I want you to appreciate that once the chromosomes have fused, there's no memory of the original break site. So any random breakage then will then generate an asymmetry, meaning that in one daughter cell, you're going to wind up with amplification of the A segment, and in the other daughter cell, you'll have a reciprocal copy number loss of the A involving the A segment. And I hope you can also appreciate that if you then iterate cycles of these breakage, fusion, and bridging, you will exponentially then amplify the A segment. So you'll go from 2 to 4 to 8, 16, etc. So although it's widely been assumed that BFB cycles, largely due to telomere crisis, are major sources of genome instability in cancer, there's been kind of a surprising observation in actual cancer genomes, which is that the very simplest step copy number pattern with inversions that's expected from the classic BFB model is in fact not that commonly identified on its own in cancer genomes. Instead, typically there's much more extensive rearrangement that's typically interwoven with the sequence signatures of the BFB cycle. And what hasn't been clear is whether this means that the BFB cycles themselves are more complex than previously supposed, or whether you have the simple pattern which is then obscured by subsequent rearrangement. And I'm going to argue for you for the first model that the BFB model merits a revision because we think that the cycles themselves are far more complex and inherently interwoven with Chromothripsis. And the first clues to this came from cancer genome data from Peter Campbell and Christine Harrison that showed the co-occurrence of Chromothripsis and BFB cycles in some cancer genomes, which has been replicated in some experimental systems more recently. But what was not really clear is whether the association between Chromothripsis and BFB cycles is really meanings that they're mechanistically coupled events or whether these were special examples. Okay, so now I'm going to go through and summarize data on the mechanism of BFB cycles, starting with how the bridges are broken. Then I'll talk about damage that accumulates in interphase. Then I'll talk about damage in mitosis. And then we'll address the long-term consequences of bridge formation and breakage. I'm first going to show you a movie of the breakage of a bridge, and you'll see the green, the bridge is shown here in green, and you'll be able to see actin in the cytoplasm in red. And the point of showing this movie is that you're going to see a protrusion of cytoplasm along the bridge, and then you'll see what appears to be pulling of the bridge with this section going taut, this section going slack. The bridge will break. You'll see some membrane ruffling, which is characteristic of the loss of abrupt loss of tension. So the point of showing this movie is that the bridge breakage process looks like it could have a mechanical component. And I'm not going to have time to go through our data, but we have a lot of independent data supporting the idea that cytoplasmic actin contractility, like the contraction of a muscle fiber, can promote, and indeed in our systems is absolutely required for the breakage of chromosome bridges. For example, if we let the bridges form and then inhibit actin assembly, block it with a small molecule, we can completely block the breakage of chromosome bridges. Okay, so next we wanted to address the question of what are the immediate genomic consequences of forming and breaking chromosome bridges. And if the bridge breakage is mechanical, you would predict that much of the initial breakage would be simple patterns of genomic alterations, one or perhaps a few chromosome breaks. And overall, for 75% of the cases, that was true, but there's a lot of interesting detail and complexity that I'm now gonna just quickly review. So first of all, we saw, this is just a reminder here, here's a fused sister chromatids, here's the random break that breaks the bridge, and you'll see the prediction is that the daughter cells, one daughter should have a copy number gain, the other daughter cell should have a reciprocal copy number loss, and that's true in 21 pairs of cells that we studied. And some of those breaks were simple breaks, as you might imagine from mechanical force-mediated DNA cleavage. However, we saw two other interesting classes, and the first was a pattern of relatively simple breaks, but a small number of breaks, not just one clean break, but a number of clustered breaks in the area, and oftentimes the break ends were recombined in a sort of mini version of Chromothripsis. This pattern was interesting in a number of ways. First, a very similar pattern has been described as being frequent in cancer genomes from the PCOG study recently published by Peter Campbell. That's the pan-cancer analysis of genomes. And they call this phenomenon so-called local jumps, and we would propose that bridge breakage could at least be one mechanism to explain these local jumps. Secondly, this raised the possibility that mechanical force wouldn't just give you clean breaks, but could give you small-scale fragmentation in and of itself. And to test that idea, we decided one of the simplest first things we could do would be to mechanically break bridges ourselves. And we did that with a microcapillary that's shown here. And this is a cell, I'm sorry. This is a cell with a chromosome bridge, and we're gonna then watch the capillary come in and break it, and then we're gonna sequence the daughter cells. Okay. So here's the capillary, it's stretching the bridge breaks, and now we can grab these two daughter cells and sequence them. And what we found was really striking. We not only got the predicted, numbers of examples of clean breaks and reciprocal copy number change, but we also, with mechanical force, recapitulated these small-scale fragmentation. So mechanical breakage of chromosome bridges can lead to small-scale fragmentation. And overall, because mechanical forces are so affected by the cell's microenvironment, I think this raises really interesting questions about how the microenvironment in developing tumors might impact the frequency of events like chromothripsis. So that explains about 75% of the immediate consequences of bridge breakage, but that left us with another 25% that had obvious chromothripsis. And the chromothripsis was specifically occurring at the regions that we could infer was in the chromosome bridge. And we wondered what the basis for this could be. And I'm going to propose a hypothesis. I told you that micronuclei undergo very poor DNA replication. And I'm going to make a proposal that micronuclei and bridges are very similar in terms of DNA replication. And this low-level starting amount of Chromothripsis might originate from abnormalities in DNA replication. And one reason that we thought about this came from this study from the group where we looked at the nuclear envelope assembly defect on lagging chromosomes and in the subsequent micronuclei. So this is an anaphase figure. Here's a lagging chromosome. And the key point out of this study is that the nuclear envelope assembly is highly abnormal on lagging chromosomes, and the very same defect occurs on chromosome bridges. So you can see on the lagging chromosome is that some nuclear envelope proteins assemble very normally on micronuclei, but other nuclear envelope proteins, particularly the nuclear pore complexes, are strongly deficient in their assembly on the lagging chromosome. And as I said, the same thing is happening on the bridge. And in other data that I don't have time to go through in this paper, we were able to show that this defect in nuclear pore complex assembly led to defects in nuclear transport, which then created an abnormal nucleoplasm with abnormal levels of DNA replication and repair factors, which we could show through experimental manipulations was actually causing the defect in DNA replication. So this is, the nuclear envelope assembly defect is sort of the original sin, it appears, in this process that sets up defective nuclear envelope stability and also defective DNA replication. And consistent with this idea, we found that the chromosome bridges undergo markedly deficient DNA replication, just like the chromosomes in the micronucleus. And moreover, we could validate this not just with imaging, but also by copy number firm single-cell DNA sequencing. How would replication errors then give you a Chromothripsis pattern? And this slide is just to remind you that when Chromothripsis was first identified, there was quite a debate about the underlying mechanism. So one idea was that the chromosomes were fragmented and randomly religate together, which was proposed by Campbell and we think validated in the single-cell experiment from us in 2015. However, there is an entirely different model of a Chromothripsis-like pattern originating from abnormalities in replication. This is the so-called microhomology-mediated break-induced replication model advanced by Lufsky, Ira, and a number of others. And the idea is that you could have a chromosome break with a break end where the replisome essentially goes rogue and starts jumping over the chromosome or to occasionally other chromosomes and starts copying one bit here, another bit here, switches strands, etc. And you can see how this could also lead to a very similar pattern. Initially, I think these models were considered to be competing, but if you think about it, they're actually non-exclusive. And I'd like to propose to you that they may in fact be linked, and that in an abnormal nuclear environment like a micronucleus or a bridge, you could wind up with both abnormal replication and fragmentation and ligation. And some evidence supporting the idea that concomitant with fragmentation, you could have abnormal replication actually came from analysis in our 2015 paper, because we could see examples where in a long-range rearrangement junction, we curiously could see little 200 or 300 base pair fragments from typically from the same chromosome, but from anywhere on the chromosome, either arm, that were essentially strung together in a pattern linking the long-range rearrangement. And of course, we didn't have direct evidence for this, but the simplest idea would be that this was an example of replicative copying of these small segments that would lead to this rearrangement pattern. When we first noticed it, we didn't know how significant or important or common this would be, but we now have some evidence that it will be very common. And that started with our sequence analysis of the chromosome bridges, where we essentially saw a very similar pattern. And that's cartooned in the colored diagram here, where you have a long-range rearrangement junction from here to here, and then stuffed into this rearrangement junction are little, approximately 200 base pair fragments from all over this region that underwent chromothripsis. We now have better resolution and deeper sequencing, and what we've now appreciated is that all of these little segments are coming from discrete breakpoint hotspots that are color-coded here within the region undergoing chromothripsis. So what's happened here is that you have breaks, and little bits of the break area are all getting copied together in this very interesting and unusual pattern. Now, the first thing that we thought was that this would be some artifact from single cell analysis, but that's not the case, because we grew clones of these cells and we could see the pattern by conventional short read sequencing. The next thing that we worried about was that it might be, something that we were not doing a correct job in linking up all the short reads, but that was not true, because we have unpublished data from PacBio sequencing on the clones that we can essentially confirm everything that we saw by short read sequencing. Now comes the question of, is this really important and relevant in cancer? And the answer to that seems also to be yes. I mentioned this phenomenon to Peter Campbell, and he kindly hooked us up with Tom Mitchell from his lab, who is studying chromothripsis at the characteristic site in renal cell carcinoma. And with long read nanopore sequencing, they've essentially seen the same kind of pattern. And there are now a number of other studies that see this pattern in a different context. So we believe this pattern is most simply explained by DNA replication abnormalities, but we don't have the final answer on that. We have a number of indications that this is going to be a very common pattern in cancer genomes that's going to tell us something important about the initial replication stress that occurs in cancer genomes. So this will be an important topic to follow up and is an ongoing story. Okay, but just to sum up this part, I want to propose to you that bridges and micronuclei, even though they look very different, may actually have more in common than we thought. They acquire few nuclear pores. They likely both have a similarly defective nucleoplasm. They both undergo poor DNA replication, membrane rupture, DNA damage, and a low starting frequency of chromothripsis. Okay, so the bridges and intact micronuclei start out with low-level defects, but then things get worse. And I'm going to show you some data that in the next mitosis, the under-replicated chromosome segments that were either in micronuclei or chromosome bridges undergo massive DNA damage coupled to a burst of unexpected DNA replication. Okay, so we wanted to test what would happen to micronuclear chromosomes independent of events that would happen when the nuclear envelope breaks in interphase. And to address this, what we did was to make micronuclei, by live cell imaging, identify cells with intact micronuclei that went all the way through interphase up until the start of mitosis with intact micronuclei. and then arrested those cells in G2, pulse labeled with EDU, and then stained them. What you can see here is that, as expected, these G2 micronuclei undergo little DNA replication, little DNA damage, and as if we sequence these cells, about 10% of these cells had Chromothripsis, similar to the 25% that we saw in the newly broken chromosome bridges. But a really interesting and exciting thing happened after we released these cells from the G2 block and came back 20 minutes later in mitosis. And what you can see here is that there's massive amounts of DNA replication, specifically on the micronuclear chromosome, which is coupled to very extensive DNA damage as assayed by gamma H2AX. If we did this with cells with broken chromosome bridges, we got essentially the same kind of result. And importantly, our single-cell sequencing data really supports the idea that passing through mitosis and acquiring this kind of damage really promotes chromothripsis. Because as I told you, the cells with in G2 have only 10% of the intact micronucleated cells with chromothripsis. But if we let those cells pass through mitosis, in nearly 90% of the cases did we see then chromothripsis. So passage through mitosis promotes chromothripsis. And then for the cells with the broken bridges, things actually get even worse. I'm not going to have time to discuss this in detail, but we have evidence that the stretching of chromosome bridges may compromise the function of the centromeres. I'm happy to take questions about that after the talk. And these compromised centromeres lead to abnormal kinetochore assembly and then the subsequent partitioning of the broken chromosomes into micronuclei, which, as you can obviously now infer, will then generate further cycles of chromothripsis. Finally, we wanted to address the long-term consequences of chromosome bridge formation and breakage. And to do this, what we did was to use CRISPR to generate specific chromosome bridges containing chromosome 4, watch those bridges form, watch them break, isolate the daughter cells, and instead of sequencing the daughter cells immediately, we then grew them into populations of cells that we could then analyze by bulk sequencing, single-cell DNA sequencing, or isolate subclones, followed by, again, bulk sequencing of these subclones. And the conclusion from this analysis is that the single cell division error, the formation of the bridge, as we predicted from the results I just described, leads to extensive amounts of ongoing genome evolution and the rapid acquisition of highly complex genomes. And you can see this in the bulk populations because what you're looking at here, the two colors show the maternal and paternal haplotypes. And what you can see here is that the copy number for the chromosome bridge is present in non-integer copy number levels, indicating that there is subclonal heterogeneity in this population, which we could then confirm here by single-cell sequencing. And then when we identified subclones, we could again confirm the ongoing evolution. This is an example where 3 subclones all shared one break, but then had multiple breaks that were private to this lineage, which really establishes that the blue breaks in dashed lines happened after the orange break. So to summarize, I want to propose to you that actomyosin contractile force, by promoting the breakage of chromosome bridges, can create a surprising impact on compromising the integrity of the genome, raising very interesting questions about the microenvironment of tumors and how it interacts with the genome. Again, in work I didn't have time to discuss in detail, we think that these forces similarly compromise the epigenetic mark for the centromere, creating micronucleation. And I've reviewed for you a cascade of events that are shared by micronuclei and chromosome bridges that promotes chromothripsis and extensive ongoing genome evolution. And all these add up to show that single bad mistakes in cell division, if they give rise to chromosome bridges or micronuclei, can rapidly generate much of the complexity of a cancer genome. And then finally, I just want to come back to the original question, in that we have much to do to really understand how normal genomes evolve into cancer genomes, but I do want to suggest that there has been quite a lot of rapid progress in the field from many groups and from many angles, from cancer genome sequencing to cell biology and single cell genomics. And I think we now have a bit of a clearer understanding of how this process is occurring, though we have to analyze it in more detail in cancer relevant models. Here, this work has been involved many people in my lab and many collaborators. Early on, I want to highlight Matthew Meyerson's group that collaborated with us initially on the single cell genomics, and for the work on chromosome bridges was all driven by a very talented postdoc, Neil Umbright. All of our bioinformatic analysis is a very close partnership with Chengzong Zhang, who's an assistant professor in biomedical informatics at Harvard and actually a former postdoc in the lab. And many others contributed to the Bridge project here. And with that, I'll stop and take your questions, and thank you for your attention.